So please welcome our next speaker, Dr. Schneider, City of California, San Francisco.
All right, hi everyone. First of all, thank you to our amazing organizers for putting the symposium together. I'm excited to describe my postdoctoral work, which is going to be the de novo design of functional proteins and what we can learn in terms of protein structure, function, and recognition by the design of these classes. So why should we design proteins? Well, from a fundamental standpoint, if we truly understand protein folding, and by that I mean the biophysics, the biochemistry, and the thermodynamics that govern protein folding, we should be able to design proteins from scratch. And if our understanding of these is truly atomic in nature, then we should be able to springboard from that to the design of proteins with new and unanticipated functions, such as protein materials, enzymes, and drug delivery vehicles, just to name a few. So my work in the DeGrotto lab is going to be addressing a few open objectives in the field of protein design. The first is the precise control over protein shape, specifically when dealing with large heteroprotein peptide complexes. And the second is the consideration of protein dynamics, or more specifically, the design of perfectly fine-tuned protein complexes. So we'll start with the nanopore project, our precise control over protein shape. So for those of you who aren't familiar, protein nanopore-based sequencing is based on the accurate measurement of a flow of ions through a membrane-embedded pore. The current varies as the DNA passes through the pore itself, and we can read that variation to then go ahead and sequence the DNA that passed. So this is a membrane pore embedded onto an artificial membrane. There is a current that passes through the pore itself, and the bases that pass are going to disrupt the current. We read that disruption and then use it to fingerprint the bases that had passed through the pore. Now, there are many different protein nanopores that have been utilized both in industry and in academia for nanopore-based sequencing, most notably alpha-hemolysine and MSPA. You can see a few examples here. They differ primarily in their shape and the lumen architecture, particularly the constrictions to the lumen themselves. And these are very important. As I mentioned, we need a very specific, very silent baseline current so that any changes to the current itself is going to come from the passing of the bases to the pore. So Oxford Nanopore, which are our amazing collaborators on this project, have been working for about a decade to develop the CSGG-based pore as a pore for nanopore-based sequencing, for commercial nanopore sequencing. So CSGG is part of the E. coli curly biogenesis system. It is a non-ammeric complex. So this is 9 copies of CSGG, the protein that you can see here in green. And Oxford Nanopore, again, have been developing this pore as a commercial nanopore for about a decade. This is a highly stable complex. In 2020, Oxford Nanopore, together with a group in Belgium, solved the cryo-EM structure of CSGG with its natural binding partner, CSGF. This is the N-terminal part of that protein, which you can see here in orange. Now, what they found very interestingly is the addition of CSGF, or the N-terminal peptide of CSGF, to CSGG results in protein nanopores with high homopolymer read accuracy, which is a big want in the field of nanopore-based sequencing. However, while this complex, CSGG alone, is highly stable, the CSGG-CSGF protein nanopore is relatively unstable. And you can imagine that the formation of this type of complex, a heteroprotein peptide complex with 18 subunits, is going to require high specificity and complete cooperativity between all the subunits to form. So if you look into the protein lumen architecture of the CSGG-CSGF pore, you can see again the constriction coming in green from CSGG and the second constriction to the pore in orange from CSGF. What is interesting is that Oxford Nanopore found that the C-terminal region, what you can see here, of CSGF is the primary reason for the instability of the CSGG-CSGF complex. You can begin to understand this is a relatively unstructured, possibly quasi-polyproline 2 region, but they found that when they systematically shortened this unstructured region, what they hoped would be that it would stabilize the structure. However, any shortening causes the CSGF peptide to fall out of the pore itself. What they also did was they tried to heavily mutate the CSGF-based pore, and no mutations really significantly stabilized the nanopore itself. So that's where we came in. They asked us to design this highly stable complex with minimal dynamics and try to address the floppy C-terminal region itself. There's a few ways we could do that. The first was our biochemical approach. We used suffix-based chemical cross-linking to covalently stabilize this 280 kilodalton 18 subunit complex. I don't have time to get into exactly what we did, but we patented this technology, and this is also published last year. Please come talk to me about it later if you're interested. But what I'm really excited to talk to you about today is our de novo design approach, or how do we stabilize this protein complex by the addition of another protein subunit. So as I mentioned, from the work of Oxford Nanopore, we knew that we can mutate this peptide and we can shorten it. So our idea was, how about we add on to it? And as you can see, we moved from what you can see here on the left, this is CSGG in green, wild type CSGF in orange, to our de novo CSGF, which is what you can see here in pink. Now, some of you may be thinking, well, you're just adding another helical hairpin. How challenging could that be? Well, I probably wouldn't be talking to you today about it if it was easy. And the addition of this helical hairpin with atomic level precision within the very, very confined space of the CSGG base pore is something that really puts our protein design capabilities to the test. We're not designing in a vacuum. I can't diffuse any structure to be as long as I want, as tall as I want, as big as I want. Oxford Nanopore spent a decade developing the CSGG base pore. We need to perfectly tile that surface of CSGG. In addition, we need to make sure that what we're designing has absolute cooperativity and specificity. We need these to be rocks. And we need to have a precise pore lumen architecture that allows for the DNA and RNA to still pass through the pore itself. What this actually boils down to, if we think about it, is balancing the cooperativity generating homotypic interactions between the CSGF monomers that we will be designing, as well as the specificity generating heterotypic interactions between the CSGG interface, as well as our de novo CSGF. So how do we go about doing this? We're going to rely on the wisdom of evolution that we're going to be able to pool by searching through the PDB. So just to orient you, we're looking at 3 CSGG-based monomers that you can see there in green, 3 CSGF-based monomers that you can see there in orange, and our queried region is what you can see there in red. This is the helical part, the midsection of CSGF. So we develop an algorithm that allows us to first identify all the red helices in the PDB. For an analogy, that's going to be my fist here. This is that helical region of CSGF. And we ask the PDB a relatively straightforward but very powerful question. Find all the good geometric matches within a given distance in RMSD to those helices. So find me all the good geometric matches to my fist. But I only want those hits that face towards the lumen, and that's where that mask comes in gray. Don't give me hits that face towards me or towards CSGG, but give me only the hits that face toward the lumen itself. Now you can imagine that there are quite a few hits that come up with this type of search, and our algorithm is going to base the hits on their prevalence. When I say prevalence, I mean how many times they have come up over and over again in the PDB, which is how many times they have come up over and over again in evolution. And the idea is that if there are many sequences that can fold to give me that same structure, that same good geometric match, then that piece of secondary structure is going to be highly designable, which is very much needed for this type of design project. So we middle based on prevalence, based on designability. That's our first and very important metric. We can then whittle these down based on secondary structure and orientation, but very, very importantly, and this is where the secret sauce of the algorithm and the design approach comes in, we need to make sure that we account for symmetry. Both my CSGF symmetry mates on each side, but more importantly, or equally importantly, my CSGF symmetry mates across from me. I need to be designing the pore lumen very accurately. I need to make sure that I have a baseline current that is completely silent and that we still leave enough room within the confines of CSGG for the DNA and RNA to pass through the pore. Once that has been done, we loop the 2 secondary structure units, which is an art in and of itself. We apply symmetry. To make a very long story short, we design 2 design generations. What you can see here in green and orange is the wild type pore. This is a 20 angstrom in diameter pore. You can see our first generation here in pink, which is a 16 angstrom diameter pore, and our second generation, which is a 12 angstrom diameter pore. Importantly, we only generated 5 designs per each one of these generations. This is not the issue where you have, hundreds or thousands of designs that you can check where you're doing directed evolution. We have to design these with pinpoint accuracy, or else you get the whole R&D department of Oxford Nanopore pretty upset. So they differ not only in the size and the diameter, they also differ in the lumen architecture, as well as the vertical height that they stick up out of CSGG. So once those 5 designs per generation have passed our criteria, we hand off these design stocks for Nanopore and they incorporate them in their sequencing devices. They sequence for 30 minutes and then they look to see how much or what percent of the pores are actually CSGG and CSGF, which is what percent of the pore are only CSGG alone. So for the wild type complex, only 41% after 31 minutes is the correct complex that we're looking for. Whereas very gratifyingly for our designs, we're at almost 80%, so almost a twofold increase. And this is something that is extremely important if you're thinking about pores that are being manufactured on a global scale. So not only are they very stable, but they also contribute to the accuracy of the overall reads. You can see here actual reads from nanopores that are either CSGG alone or from our design CSGF. And really excitingly, recently we solved the cryo-EM structure of our designs. So everything that we had thought in terms of design consideration, I only talked to you about backbone choice, but there was a lot of work that was done in terms of sequence. All our considerations about how this would work and how we would perfectly balance those homotypic and heterotypic interactions are now actually design principles because we have corroborated our design concerns with the cryo-EM structure. So moving forward, we are really excitingly now working on the design of nanopores for protein sequencing. This is really the next frontier in nanopore-based sequencing. And we are also designing nanopore-based sensors. So that was our first open objective. How do we precisely control an atomistic level of protein shape? And our second is the consideration of protein dynamics. Or how do we specifically design proteins with fine-tuned stability? What we're designing here are masks for bioactive peptides. I'm talking here about primarily two families of bioactive peptides. These are antimicrobial peptides and cell-penetrating peptides. These are amphiphilic basic peptides, so they have basic residues that are functional. They readily interact with membranes. Antimicrobial peptides with the bacterial membranes, and cell-penetrating peptides with mammalian cell membranes. They readily interact with these membranes, but this is also the bane of their clinical use because they lack specificity. Our idea was, how can we structurally and electrostatically mask these bioactive peptides within a protein mask, and then only conditionally release this protein upon a predetermined cue? In this instance, we're using protease cleavage. So basically what we're asking is, how do we design a protein complex that is stable enough to shuttle a bioactive peptide where in the pre-Q state, but not too stable or perfectly balanced so that it readily and rapidly releases this bioactive peptide in the post-Q stimulus. So the first example that we had are cell-penetrating peptides. I'm showing you an actual prototype. The cell-penetrating peptide here is in orange, our mask is in green, and our protease cleavable loop is there in yellow. And as our model cargo, we chose an M-cherry fluorophore. And the way we envision these masks working is such. We are first going to design the full complex, again, the cell-penetrating peptide, which is being masked by our de novo mask, the protease cleavable loop that has this M-cherry cargo. Following protease cleavage, we need rapid and reliable release of the cell-penetrating peptide from the mask itself. To do this, we need to make sure that our K-dis is over the IC50. We need to make sure that we actually deliver the peptide in a concentration that allows it to readily interact with the membrane. We also need to optimize loop length. As I mentioned, we're using protease cleavable loops. We can't design loops that are very, very long to allow for proteases to actually cleave the loops because we know that there's an inverse correlation between loop length and protein stability. So these need to be fine-tuned as well. And to jump forward, I'll show you some experimental results from our first prototype. So here you can see HeLa cells. In blue is nuke blue staining. These are live stained cells. These are HeLa cells that were incubated with either the complex alone or the complex that was protease cleaved. That's what you can see here. And any red that you see comes from the M-cherry cargo. So you can see the distinct difference of HeLa cells that were treated either with the complex itself or with the protease treated complex. So this is an example that we did with cell-penetrating peptides. We've also done this with specific endosomal escape peptides as well. And I hope that you can appreciate the modularity of this design approach. So we have been doing this, I've shown you for cell-penetrating peptides. Excitingly, there's been a wide group of people in the lab who have joined me working on antimicrobial peptides. We're also working on peptide hormone cages as well. The Baker lab also has amazing work that is not all too dissimilar from what we're doing here, but for cytotoxic proteins. And in terms of future plans for this part, we're now working on in vivo delivery, which is really exciting, the introduction of targeting moieties, and we're working on new scaffolds because they can't all be helical bundles. And with that, I will conclude. I will thank Bill and my amazing collaborators, both in the lab and Oxford Nanopore. I will thank my funding and thank you all for sticking with us to the end here. I'd be happy to take any questions.
Thank you so much, Lee. Yeah, unfortunately, felt the lack of time. We don't have time for live questions, but guests and moderators and panelists, please feel free to write your questions in the chat and Lee can answer them. And thank you so much. Lee,